I managed to get a little timer to just tell me what the time is so that I can keep track.
We can tell you as well.
Yeah. Okay.
Okay. Awesome. Jen, I think we can start while people trickle in. It usually takes a few minutes for people to join in.
Whenever you want to go, up to you.
Okay, I think I'm ready. Okay, awesome. Thank you so much, Ajay, for joining us today and everybody else for another session of Cell Migration Seminar. We are really delighted to have Ajay today. Dr. Ajay Chitnis got his medical degree in Mumbai, India, and then he came to US to do his PhD at University of Michigan, Ann Arbor. From his thesis with John Kuwada, he described how early neurons in zebrafish brain extend exons along specific pathways to establish a simple exon scaffold that guides the migration of later developing neurons. He then moved to Salk Institute as a postdoc, where he worked on Xenopus embryos with Chris Kittner and discovered the lateral inhibition mediated by delta notch signaling pathway. He did another short postdoc in Boston with Wolfgang at Mass General Hospital, where he identified zebra mutants with an aberrant pattern of early neurons. Since 1997, he moved to Bethesda NIH, where I'm as well, to establish his own lab. And his lab has been focusing on zebrafish to understand how interactions between cells determine the self-organization of various aspects of neural development. More recently, his lab has been focusing on remarkably accessible and relatively simple zebrafish posterior lateral line Primordium, which most of his recent work has been about. And his lab is awesome in like live imaging with a lot of genetic and molecular chemical manipulations, and they try to understand how interaction between cells coordinate cell fate specifications and morphogenesis, and also very specifically collective cell migration, which I think he's going to be talking about. With this, I would really like to welcome Ajay. Thank you so much again for joining us, and we're delighted to have you here.
Alrighty, thanks so much, Ankita, for the invitation. It's really, I've enjoyed the seminar series right from its beginning. So I'm going to start my talk today with acknowledging all the people in my lab that have made this possible. But I'm going to start in a rather unusual place with this mosaic, which I find fascinating because it's got multiple patterns nested one within the other. So, you see a dome over here made up of small mosaic pieces. And at some large scale, you can see that it's made up of these giant polygons. There are pentagons and kite-shaped things and this bow tie-shaped thing. But if you look at a finer resolution, you can see that the same patterns repeat themselves at a much smaller scale. And then one of the most interesting things about this particular mosaic is that each of these patterns that I'm focusing on, whether it's the pentagons or these bow tie shaped things, are themselves not actually, the mosaic pieces don't actually have these shapes, but these shapes emerge by the juxtaposition of larger mosaic pieces, which are of a completely different shape. So why am I showing you this mosaic? To me, this mosaic is like a metaphor for what I find most fascinating about living systems, where there are nested levels of self-organization with recurring motifs, molecules, and emergent behaviors at multiple scales. So it's with this in mind that our lab has been studying the lateral line primordium for last 10 years or so. And we try to understand its organization at multiple scales to try to understand as a sort of an example of how this process happens during animal development in many different contexts. So the posterior lateral line primordium is this group of about 100 cells that migrates from near the ear to the tip of the tail, periodically making and depositing neuromass, which are these sensory organs of the lateral line, which allows the fish to detect the pattern of water flow over the cell over its body surface. And if you look a little closer, you can see that this migrating primordium is moving along this very well-defined line that is the horizontal myoseptum. And so this is what we're looking at here is a sort of a lateral view of the migrating primordium. And instead, if you were to look from the top, look dorsally, you could see that the primordium is migrating, sandwiched below with the muscles below and the skin above. More recently, we've been looking at the primordium with even higher resolution. And what looking at this resolution allows you to see is that this structure is actually composed of a variety of different cells that are working together to give you the emergent behavior and morphogenesis within the system. So work from number of labs has already shown previously that leading cells tend to be a little less organized. Whereas the trailing cells are polarized, they become tall and become apically constricted to form these little epithelial rosettes. At the same time, at the center of these rosettes, a cell becomes specified as a hair cell progenitor. And just prior to the deposition of this rosette, that progenitor is going to divide to give you the first two sensory hair cells of the neuromast, which will then go on to mature to give you many more hair cells. More recently, if you take a cross section through this primordium, you can see that some of these cells have organized to form these beautiful rosettes. But more recently, our work had focused on the discovery of these superficial cells, which we had simply not realized were there and not really paid attention to. And some of our recent work showed how these superficial cells, in fact, extend processes against the overlying skin. And you can see that they extend these beautiful lamellipodia against the skin surface. And this, as I will discuss later, allows and facilitates the migration of the primordium. So what we see is that the primordium has a diversity of cells and subcellular structures that are formed and maintained at various scales. And really what our goal has been to try to understand the regulatory networks that coordinate the formation of these structures and how interactions between the regulatory networks, the structural components, and mechanics all work together at different scales to make the system work. So my talk today is going to be in sort of 4 parts. In the beginning, I'll give you a sort of an overview on how polarized Wnt and FGF signaling coordinates many aspects of neuromast formation and collective migration. I'll then talk about the regulatory networks that create a sort of a pre-pattern that defines where these rosettes are going to form within the migrating primordium. And then I will talk about how, despite the fact that there are these networks that define where the rosettes will form, ultimately the formation of rosettes, migration, it's all a matter of mechanics, and the interactions between mechanics and signaling in the system can influence the size and number of rosettes formed within the migrating primordium. And in the last part of the talk, I'll shift to what we have begun to learn by looking at the behavior and shape of dynamics of the shape and behavior of individual cells, with the goal ultimately of trying to understand how the behavior of individual cells is going to contribute to function, morphogenesis, and migration at the tissue scale. So let's start with our overview. So as you all know, the primordium migrates along this path. And as I have mentioned earlier, the leading cells tend to have a more sort of a quasi-mesenchymal morphology, whereas cells in the trailing part of the primordium sequentially reorganize to form these epithelial rosettes. And as these epithelial rosettes are formed, a cell at the center of this rosette will become specified as a sensory hair cell progenitor, and its surrounding cells will become specified as support cells. So work from a number of labs has already shown that a lot of what the coordination of both the formation of these rosettes, the specification of hair cell progenitors, and migration is all coordinated by 3 important signaling pathways: the FGF, Notch, and Wnt signaling pathways. So what are these interactions that allow these rosettes to form sequentially from the back to the front? So initially, the primordium is dominated by Wnt activity. It is broad. And as the primordium begins its migration, that wind activity becomes polarized in the primordium such that it is highest at the leading end and least active towards the trailing end. How migratory behavior facilitates the formation of this gradient of wind activity is not clear at this point, but we know that migration seems to be important for establishing this gradient of activity. Now, wind activity within the primordium, there's three important things that are shown over here. On the one hand, it drives the expression of various factors that promote its own activity. The second thing that it does, it drives the expression of these FGF ligands. But these FGF ligands are not able to activate FGF signaling within the cells that produce the FGF, because the third thing that the wind activity does, it drives the expression of factors that inhibit the effective activation of the FGF receptors. So these FGFs that are produced in response to wind activity are delivered towards the trailing end, where because there is weaker wind activity and less inhibition of the FGF receptor, the FGFs are able to establish a FGF signaling center. Now, what's interesting is that the signaling center, the pattern of FGF activity is center biased, as I have shown with this blue shading over here. And this center biased FGF activation leads to the expression of center biased 801 expression. So 801 is a transcription factor that gives cells the potential to become hair cell progenitors. However, lateral inhibition mediated by NOT signaling allows each of these cells to try and inhibit their neighbors from becoming an hair cell progenitor. And as a consequence of lateral inhibition, 801 expression becomes restricted to the central cell, which becomes specified as a sensory hair cell progenitor. This cell now becomes a new source of both delta and FGF, and delta and FGF activate notch and the FGF receptor in their neighboring cells, and this helps to stabilize the formation of epithelial rosettes around the central hair cell progenitor. Now, once this FGF system has been activated, it in turn begins to inhibit the wind system. And so this wind system that was initially very broad becomes progressively inhibited and restricted to a smaller leading part of the primordium. And as the wind system becomes restricted to a leading part, conditions are such that now you can establish a new FGF active domain, which will seed the formation of yet another neuromast. So this process continues, starting from the trailing end, and one by one, you start getting protoneuromasts forming progressively closer to the leading end, with the wind system progressively getting squeezed to a smaller and smaller part in the leading domain. As the system, as the cells proliferate and the primordium becomes longer, eventually the trailing cells lose their capacity for migration and they are deposited. If they had been incorporated in these epithelial rosettes, they are deposited as neuromasts. If they are not incorporated into this rosette, they are deposited as interneuromast cells. So one of the things, so this sort of brings me to the first puzzle that I want to talk to you about. One of the things we discovered is that as the wind system progressively shrinks towards the leading end, it turns out that as neuromasts start getting deposited, the entire length of the primordium also begins to shrink. And when we measured the length of the wind system compared to the length of the entire primordium over time, you can see that the wind system progressively shrinks. The primordium initially grows in length as more cells are added, but once neuromasts start depositing, it also begins to shrink. And if you look at the ratio of the size of the wind system to the length of the primordium, it turns out roughly it is always at a fixed proportion. It happens to be 60%, but the interesting thing is that there is a sort of a fixed relationship between the size of the wind system and the length of the primordium. So this was a puzzle that we were wondering about. Why what is it that links the length of the primordium to the size of the leading wind system? And one answer to this question, we believe, comes from understanding the mechanisms that determine the migration of the primordium. So you've already heard a beautiful talk from Holger um, a few weeks ago, where he described how the degradation of chemokine CXCL12A by the trailing cells allows the formation of a self-generated gradient that allows the primordium to migrate along this well-defined path defined by the expression of CXCL12A. So what we showed a few years ago, so it was already known that in addition to the primordium migration following this path defined by CXCL12A, that migration was also dependent on FGF signaling. So what we also showed was that these FGFs that are secreted by leading wind active cells not only are delivered to the back where they help to make these rosettes, but they serve as migratory cues for the directed migration of trailing cells. So I'm not going to describe the experiments that led to this conclusion, but one of the things we were able to do was to cut the primordium and show that in a situation where you've cut the leading part from the trailing part of the primordium, trailing cells move towards the leading cells, and that this behavior, it turns out, is dependent on FGFs secreted by the leading cells. So what we have—
sorry to stop you quickly. There's a clarification question from Robert Insal asking, when you
Sure.
say that the neuromast precursor cells lose their capacity for migration, do you know
Yes.
this, or do you just observe that they stop moving?
I would say it's more accurate to say that we observe that they stop moving. We don't completely understand all the reasons why they stop moving. I think it's actually kind of a more complex process, but let's just say they lose their capacity to migrate, we think, and I'm going to suggest at least one reason why they might do it.
Thank you so much.
Alrighty. So what we think happens is that as the primordium, the cells proliferate and the primordium begins to elongate, and the wind domain, which is the source of the FGFs, becomes restricted to a leading domain, the access to these FGFs that serve as migratory cues or other wind-dependent factors that may be required for migration are progressively lost in trailing cells. And this, we believe, contributes to the loss of the capacity to migrate. And as I said, you then are deposited, and if you are already in a rosette, you will be deposited as a neuromast or as an interneuromast cells. Now, what we think happens is that as the wind system shrinks, the size of the source of these FGFs is also progressively shrinking. And one idea that we will sort of explore with the computational model a little bit later is that as this leading source of wind of FGFs shrinks, the capacity to migrate is lost progressively closer to the leading end of the primordium. And as a result, as the wind system shrinks, together with it shrinks the entire primordium. So this is an idea that sort of is more like a hypothesis that whose validity we will explore with a computational model a little bit later in the talk. So before I get to that, I want to explore talk a little bit more about the regulatory networks that we think determine or help specify the spatial pattern in which the rosettes form within the migrating primordium. So I've already told you about how interactions between the wind and FGF system create the center biased pattern of FGF activity within which 801 expression is initiated and a central hair cell progenitor is specified, and then how that central hair cell the sensory hair cell progenitor becomes the source of signals that helps to consolidate the formation of a stable epithelial rosette around it. So one of the big puzzles that was really um, difficult to figure out in the beginning is that if you look at this set of interactions between the wind system and the FGF system, it's easy to understand that the wind system inhibits FGF, the FGF system inhibits the wind system, and these sets of interactions you might imagine simply by divide the primordium into two compartments, a leading compartment dominated completely by wind and a trailing compartment completely dominated by FGF. So why would it be that this interaction might actually initiate center biased a pattern of FGF activity? Well, before we got to that point, we actually looked at the pattern of FGF activity as revealed by the expression of PA3, which is a target gene of FGF signaling. And consistent with this sort of schematic that I've drawn, where the nascent neuromast is characterized by center biased FGF signaling, and then once the central cell becomes a source of FGF signaling, the pattern of FGF signaling becomes much broader. If you look at the pattern of PA3, it actually reflects these same changes. But what is it between these, with the interactions between that Wnt and FGF that allows this to happen? So the answer to this question came from the recognition that this seemingly simple set of interactions between the Wnt and the FGFs has a topology or a structure which is consistent with the formation of a reaction diffusion system that would spontaneously allow interactions between Wnt and FGF to create potentially spots, periodic spots of FGF activity. So I realized that this might be the case actually when I was listening to a talk from Shigeru Kondo, who was describing mechanisms that might determine the pattern of pigment patterns on the surface of the fish. And he was describing how if in any situation where you have some system A that inhibits B and B that inhibits A, but A produces some factor that can travel at a distance to promote the production of B, which is an inhibitor of A, you basically have the ingredients for this pattern forming system because you have indirect local activation coupled with long range inhibition. So this is a simple computational model to illustrate the patterning potential of such a regulatory network. And in this example, all we've done is started with a sort of a random pattern of these yellow versus blue cells, and then the cells are interacting based on this regulatory network. And what you see is that even if you start with a completely random pattern, it resolves into these patterns with either yellow spots within a blue background or blue spots within a yellow background or with stripes. And the difference between each of these situations is simply based on the strength with which the yellow system inhibits the blue system or the blue system inhibits the yellow system. And what this sort of generic model illustrates is that if you have the yellow system dominating in inhibiting the blue system, then you will resolve or evolve a pattern in which you have blue spots surrounded by yellow. So when we look at this regulatory network that had already been described for the interactions between Wnt and FGF, we realize that what we have is something very similar to this. So taking what we had learned from this sort of very simplistic generic model of this interaction, set of interactions, we developed a computational model of the lateral line primordium. So this is a bunch of cells that represent the primordium. And we implemented a computational model of this network, which is pretty much similar to what I showed you with a few additional details. And we imposed in this system a gradient of Wnt activity, which as the primordium migrates and grows in size, progressively shrinks towards the leading end. And what you can see is that simply as a consequence of this set of interactions, you can spontaneously get the formation of these little spots, center bias spots of FGF activity in the wake of a shrinking wind system. And in this model, what we have is as FGF signaling accumulates in each of these little domains, it allows the formation of these rosettes. And I'll just run the thing again, maybe. And as I had suggested in this model, what we have is when as the FGFs that are secreted by these cells diminish towards the trailing end, once they drop below a certain threshold, the cells lose their capacity to migrate and get deposited as neuromass if they were part of these rosettes, or as interneuromass if they were not part of it. And in the beginning, I had suggested that this idea that as the wind system shrinks, the size of the domain which is producing these FGFs will also shrink. And this could potentially provide an explanation for why the length of the primordium shrinks along with the shrinking wind system. So what we could do to see whether this suggestion makes sense was to go back and look in these simulations and compare the length of the leading wind system to the size of the primordium. And without this actually being an explicitly defined feature of the model, you can see that this idea is at least theoretically adequate to account for why the primordium shrinks in a manner that scales with the size of the leading wind system. So at this point, I've spent a lot of time talking about these sort of regulatory networks, how they might create a spatial pattern of periodic spots of FGF activity that will seed the formation and location of these protoneuromass, which form starting from the trailing end of the primordium. However, what we've discovered is that while there are these signaling interactions that can influence where these rosettes form within the migrating primordium, ultimately the formation of these rosettes is influenced by mechanics, including by the pull and tug of the cells on each other as the primordium migrates. And we realized the influence of mechanics on the size and number of the of these rosettes in experiments where we were playing or manipulating migration of the primordium by either manipulating the efficacy of the chemokine system or the FGF system. So what I'm showing you here are videos from experiments in which we have compromised the migratory behavior of the primordium, either by partially knocking down the chemokine that guides the primordium, or using a heat shock transgenic line where we can induce the expression of CXCL12A. In both cases, either the deficiency of CXCL12, which in this case, when you knock it down partially, the primordium hits a patch where there is a low level of the chemokine and it slows down and then it speeds up again. What you see over here is that when it slows down, these pre-existing rosettes will fuse together to form one rosette. In this particular context, the primordium is slowing down because there's so much chemokine everywhere that it doesn't know where to go, and eventually the receptors get saturated and they're no longer able to direct the migration. And in both cases, as it slows down, multiple multiple rosettes come together to form one large rosette. So looking at these results and seeing how the number and size of the rosettes can get influenced by the relative movement of the leading cells versus the trailing cells in response to chemokine versus FGF signaling, made us realize that the push and pull of these cells on each other as they migrate can influence the aggregation of cells to form these rosettes. So based on these observations, we came up with a relatively simple model for thinking about how the adhesive interactions between cells coupled with apical constriction might spontaneously lead to the aggregation of cells to form these rosettes. So in this simulation, you have just a bunch of cells that are joined together with these springs. The springs are pulling on each other. And what you see is that as the cells pull on each other, the cells come together to form large aggregates. And when the size of the aggregates exceeds a certain amount, they assume this blue color. So what you can see is simply by having cells pull mutually on each other, the system can spontaneously generate a number of aggregates of cells. And if you allow this aggregation to go unimpeded, then you start with a certain number of aggregates, but then they will slowly coalesce to form one large aggregate. But if you allow the cells to migrate, then these aggregates are pulled apart. So this, it turns out, is in some sense, sort of, we are revisiting a mechanism that I described to you earlier. So with the signaling, it was local activation coupled with long-range inhibition of these signals that allowed the periodic formation of these spots of FGF activity. This is sort of a mechanical version of the same process of local activation and long-range inhibition, where the local activation is the cells pulling each other and trying to pull their cells, their neighbors together to form an aggregate, whereas the inhibition comes from the tension generated by migration. So based on these sorts of ideas, we built another very simple mechanical model of the primordium, in which the spring strength that pulls cells together is a little bit different in the leading domain, where there's Wnt activity, versus the trailing domain where there is FGF activity, where we know that cells are more likely to come together to form these rosettes. So in this system, we basically believe that spring-like adhesive interactions pull cells together to promote the formation of aggregates, whereas migratory forces pull the cells apart, inhibiting the migration, inhibiting the aggregation. But in this system, because the migration is dependent on two processes, chemokine signaling, which dominates in the leading part of the primordium, and FGF that also contributes to the migration of trailing cells, the ultimate tension in the system is dependent on the relative efficacy of these two systems. And if you compromise the migration of leading cells, which is dependent on chemokine signaling, and the whole system collapses like a concertina, allowing pre-existing rosettes to fuse to form a single larger rosette. So while these experiments were going on in our lab, looking at how mechanics might contribute to might work in parallel with signaling systems to determine the size and number of rosettes, Damien in the lab had been exploring the behavior and movement of individual cells in the primordium. And this is what led to the study that we published a few years ago, where he had discovered that, in fact, in addition to the leading cells, which were more mesenchymal trailing cells that have this polarized structure with epithelial rosettes, there was this additional population of cells sitting on the very top that is in contact with the skin, and which extends these beautiful processes to facilitate the migration of the primordium. So he spent some time studying the behavior of these processes and showed that, as had been shown before, that FGFs secreted by leading cells provide migratory cues for trailing cells. These FGFs were also providing migratory cues to direct the polarized extension of processes by the superficial cells. So this is data that shows that if you look at the orientation of the lamellipodia that are extended by both, in fact, the basal processes of these polarized cells, as well as the protrusions of the superficial cells, that their polarization is messed up in a situation where you inhibit FGF signaling. However, the most interesting observation that he made was when he peeled off the skin and observed what happens to migration. So what he found was that when you take the skin off, the primordium immediately stops migrating. But then, as the skin grows back over the surface of the primordium, it immediately begins its migration once again. Now, if you look at higher resolution, at the processes extended by the primordium cells, in the absence of the skin, you notice something else that is extraordinarily interesting. So I already showed you that these cells normally extend these beautiful lamellipodia, but the moment you take the skin off, these lamellipodia are lost and the same cells start making these blebs. So this is in some sense a phenomena that has been described in the past, where cells move in confinement. If they are moving in a context where there is a non-adhesive substrate, you tend to see migration that is based on bleb formation. So in this context, what we do see is a switch from the formation of these lamellipodia to formation of these blebs. But in this context, the bleb formation is not in a context where it facilitates the ongoing migration of the primordium. But what you notice is that as the skin grows back, there is almost an instantaneous flip in the morphology of these processes. And as soon as they are back in contact with the skin, the blebs are lost and they regain their form as lamellipodia, suggesting that this, again, is a self-organizing process that is operating at this subcellular scale, and the form of the processes extended by the cells depends on the substrate on which they are able to extend their processes. So in a context where there is an adequately adhesive substrate, it seems that the lamellipodia can form. And when you take the skin off and deprive the cells of that adhesive substrate, they switch to having blebs, but then they switch back to lamellipodia as soon as the skin grows back. So this is something that we continue to want to try to understand. We also don't completely understand how confinement, what are the different ways in which the confinement might be contributing to the effective migration of the primordium. But at any rate, as we got more and more into trying to understand how individual cells, their shape and their behavior contributes to the migration of the primordium, we also became interested in trying to understand how we can develop new types of models where we can look at the dynamics of cell shape, size, and behavior, and how that might contribute to the emergent behavior of the primordium as a whole. So far, I have shown you models that we've made in the past. First, to see the spontaneous generation of patterns of FGF activity, or to see how interactions between the cells might lead to their aggregation. So these models were what are called agent-based models, in which each of the cells that we are, whose behavior we are trying to investigate is sort of represented just as a point in a grid where the cells are allowed to move. So while these kinds of models are quite effective at illustrating these sorts of things that we've talked about earlier, they're not at all adequate in trying to investigate the dynamics of cell shape and movement and how that might contribute to the behavior of or organization within a tissue. So there are a number of different other modeling environments that are much better at doing that. But what we have been using most recently are what are called cellular POTS models. So the critical difference between an agent-based model and a cellular POTS model is that now, instead of each cell being represented just as a point that moves around in some rectilinear space, now each of the cells is defined as a collection of pixels, and changes in the size and the shape of the cells can be represented by the movement of individual pixels that represent the cells into new positions, or their addition of pixels, or the removal of pixels representing each cell to represent either growth or in size or reduction in size of the cells. So I won't be able to go into a whole lot of detail about how um, these cellular parts models are made. We have been using a particular platform called Computational 3D to develop our models. And before I show how we have used it to model the Primordium, I'm just going to show you, a bare bones Computational model to show, in principle, how these sorts of models work. So when you build such a model, what you start with is trying to define the different types of cells that are going to be part of your model. So in this sort of simple example, I'm going to show you that we have 3 types of cells. And what then you want to do is specify the manner in which these different cell types will adhere to each other or to the other types of cells in the simulation. So you define how strong or weak the adhesion will be. Now, in this model, what the model is trying to do is trying to associate or figure out the energy state of the system. So, for example, if 2 cells that are very strongly adherent come together, then that is associated with a lowering of the energy of the system. Similarly, if the cells have a particular size or specified to have a certain size or a perimeter, if the cells change in their size to exceed that size or to become much smaller than the size, then deviation from the target size increases the energy of the system. So at each time point, what is going to happen is that cells within the simulation are going to sort of flip a pixel, move it into some new place, or add a pixel or remove a pixel. And then what the system does is it calculates the effective energy associated with that change. If the movement of the pixel of a particular cell moves it to a state which increases the energy of the system, that means it's sort of moving the cell to make contact with a cell that it doesn't have good adhesion with, or it increases the size beyond what it should be, then the energy of the system is increased, and that movement of the pixel is either not allowed or is allowed at some very low frequency. If, on the other hand, the change in the shape or movement of the cell moves it towards a state that it desires, then that reduces the energy, and that change in the shape of the cell is permitted. So at each point, the effective energy is calculated in the system by looking at the energy associated with adhesion, the size and shape of the cells, and the cells also can have things like a movement potential, which represents the ability of the cells to move in response to some influence within the tissue. So in this sort of cartoon model, what I have done is just created a sort of an imaginary system with 3 types of cells: small, medium, and large. In this model, the smallest cells have very strong adhesion to each other. And so because they have strong adhesion, it's associated with a low energy, which is, in this case, put at 2. The blue cells have a medium adhesion to each other, and that's associated with a slightly higher energy, 6. And the red cells, which are the largest cells, have the weakest adhesion, and that's represented by 8. And then, of course, we can specify the adhesion between the red cells and the blue cells, and the blue cells and the gray cells. And while I've been telling you about the differential adhesion of these cells, you have been watching what happens in the simulation. So what's interesting is that once this thing loops around again, you'll see that it starts with just a random distribution of these 3 types of cells. They all start the same size, but as the cells jiggle around and change the position of their pixels, the system progressively moves towards reducing the energy so that the red cells become bigger, the gray cells become more tightly associated with each other. And with these very simple rules, a sort of mixed bag of randomly distributed cells can self-organize to form this sort of very interesting behavior, which is very reproducible. But of course, this is just an example of the power of this modeling environment and how you can use it as a toolkit to explore the behavior of cells. What we want to see is how can we use this to study the behavior of the primordium. So what we're going to try to do is now build a model of the primordium, looking at it in the side view, where you can see that it is sandwiched between the skin and the ECM below. So we built this model by having cells represent the skin. We have cells that actually represent the ECM over which it migrates and the muscles below that. Then we have a layer of cells that represents the superficial cells. And then we have two groups of cells representing the cells in the FGF domain where they are, the Wnt domain where they are more mesenchymal, and the trailing domain where they are more epithelial. And in the case of the FGF domain, these cells which are polarized and tall are special because, in fact, these polarized cells can be thought to have distinct compartments which we could specify with different constraints. So you have an apical compartment, a lateral compartment, and a basal compartment. And we'll sort of represent the polarized cells in the trailing part as a composite of these three things. In addition, we have other little tools that we can use in this modeling environment. So you can have spring-like links that connect these cells that allow you to influence the shape and size of these cells. It also creates a sort of spring-like link between adjacent cells that can allow you to represent the process of apical constriction. So furthermore, to complete the construction of this sort of primordium model, we can have the leading cells and the trailing cells move under the influence of independent migratory forces. This representing the role of chemokine dependent migration, and trailing cells moving in response to FGF signaling. So here we have the whole system. It just starts with a bunch of squares. And at each iteration, the cells are going to sort of try to change their position. And if it moves them towards a state that is satisfying the constraints that have been put on the cells, it will adopt that shape, or it will move away from it. And based on these some very relatively simple assumptions, we can see that this very simple system is able to recapitulate many of the features of the migrating primordium. So in this system, we have the skin cells strongly adherent to each other, and we have the different parts, the cells in the different parts of the primordium also specially adherent to each other. The movement of the entire primordium in the system is dependent on the blue cells in the front, the superficial cells on the top, and the basal compartments of the polarized cells at the back. So that's the only cells that are actually only components that have migratory behavior. The red bits are the apical compartments of these polarized epithelial cells at the back, and they were initially connected with these spring-like links. And what you can see is that the cells have spontaneously come together to form these little rosettes. And because of the relative adhesion of the cells within the primordium to each other, relative to the degree to which they adhere to the superficial skin cells versus the underlying ECM, in this rather simplistic simulation of the primordium, you can see that it migrates, splitting these two layers into and migrating in between. So that's kind of fun. But does this sort of simulation of the primordium allow us to simulate specific behaviors of the primordium that we've seen in experimental context? So here's sort of the wild type situation where the primordium starts in this manner and spontaneously generates this stable pattern of rosettes. In this panel, what we've done is simulate the experiment where the leading cells whose movement is dependent on chemokines partially stalls. And as it stalls, the trailing cells, which had originally formed these four rosettes, come together to form two rosettes. And then as the migration resumes, just as in the actual system, these rosettes split apart to form a larger number of rosettes. Finally, we can also play around with the parameters that determine adhesion between these cells and show that if you significantly increase the adhesion between the apical compartments of the cells, instead of resolving to form four rosettes, they'll all come together to form two rosettes, or in this case, we've even been able to make them come together to form one giant rosette. So these models are, of course, great, and they allow us to play around with the system and see how it might behave under various circumstances. But ultimately, the value of these models is dependent on what information we can learn about the shape, size, morphology, and behavior of individual cells. So to help us with building models that are more, or testing models that are more meaningful, we are in parallel working to try to understand the behavior of individual cells. So Damien, while he was in the lab, he's now left and is in a position in Italy, had developed a pipeline for us to be able to look at the to segment all the cells of the primordium into individual cells whose behavior and migration we can now follow along the entire course of migration of the primordium. So as we develop these computational models that allow us to think about how behavior of individual cells influences the emergent behavior of the primordium. At the same time, we're learning much more about the behavior of individual cells in the primordium by directly visualizing them. And so what I'm showing you here is analysis of cells of a wild type primordium, but we have now begun to collect data of this kind under various manipulations where we know that either the size of the neuromass or the mode of migration of the primordium is altered. And we hope going forward that we will be able to put what we learned from this actual imaging with the models we built to build more comprehensive models of primordium behavior. So that's where all I wanted to really tell you folks today. I hope that in sort of this rather broad survey of what we've done in our lab, I have been able to illustrate how we are looking at the primordium at multiple scales and trying to understand how interactions between regulatory networks, structural components, and mechanics come together to try to organize the emergent organization and behavior of cells in the primordium at multiple scales. So I guess I'll stop there and take questions. I just, of course, want to make a pitch for the fact that these are all the people who did much of the work. Damien did a lot of the work with the imaging and initial a lot of the different projects in the lab. Abhishek and Megan have recently been doing much of the work with the modeling, and Abhishek has a background in mechanics, so he is helping us with developing that direction of work in our lab. Pritesh is working on another signaling system in the primordium that we've not yet had time to work on. Greg and Chongmin have been, so Chongmin actually takes care of our fish. Both and Greg is our lab manager. Both of them have been working on a very interesting project related to the role of SOX2 in the primordium, which I didn't have a chance to talk to you about. And Sana has recently joined the lab and is working on some new projects. But the important thing is many of our postbacs have recently left to go on to medical school and graduate school. And so we have both postdoctoral and postbac positions available. And so if anybody is interested in any of the types of things we talked about, please get in touch with me, and I'll be happy to hear from you.
Thank you so much, Ajay. That was such a wonderful talk. It's decades of work, and it's amazing to listen to it in one go. We have some questions. I think we can. Yeah, I'll start with Arthur's question, and I think Arthur added a question later on. I think you kind of mentioned it, but what he's asking is, in the mechanical model you presented, do the springs of the cell you talk about correspond to just the adhesion or also contractility, but also does it take care of migratory behaviors like co-attraction between the cells?
Yeah,
That's
this is actually a very interesting question, because in the initial model, which is the agent-based model, we had it sort of represented a conflation of the role of sort of adhesive interactions as well as apical constriction. But in more recent cellular POTS versions of the models, we actually have the cells apically constricting by actually reducing their size. And we also have the springs linking the two cells. And actually, we've come to some interesting conclusions, which I didn't think I had time to talk about. But it turns out that previously, I always thought that apical constriction and cell adhesion would work together. And if you increase them, you would simply get bigger and larger rosettes. But what we've discovered through our simulations is that while increasing adhesions certainly increases the size and reduces the number of rosettes, if you simply keep adhesion constant but increase apical constriction, either by reducing the size of the apical compartment or by making the spring strength that brings the cells together stronger, you actually break the system up into smaller and larger number of rosettes. And I can elaborate on that. This was sort of an outcome that was a surprise from the simulations, and we're in the process of testing the predictions of the model. But we discovered that constriction on its own, keeping adhesion constant, actually leads to smaller and larger number of rosettes.
Awesome. He actually had another question later on asking, in the POTS model, what happens if the skin is peeled off? Does the result is similar type, like your experiment of stalling of Primordium? Have you like dissected that?
Oh, we have actually, in some sense, it's a very simplified model, and the way it works, in fact, you see at the bottom of the screen another model that we made that looks at the Primordium in a different way. There's no skin over here. So in these models, they're so simple that the removal of the skin doesn't actually constrain the movement quite in the ways in which it does in the real system. So, the models are good for exploring certain things, but they're giant simplifications of a number of things. And to actually see how the different ways in which the compression by the skin might actually contribute in the real system, we might have to develop a different types of models. So in the simplistic version, we didn't specifically take that model that we've got and remove the skin and see what happens, though that's something we should do. My suspicion is that it won't quite have the dramatic effect that happens in the real system.
Awesome.
Great, thank you. Yeah, we have a question from Natalie Hussein asking if there's a correlation between the type of adhesive molecules in the Primordium and the strength of adhesion.
We don't know the answer to that. Actually, things that we don't quite understand yet is, there are a number of adhesion molecules expressed in the Primordium. Much of the attention has been in the past on, N-cadherin or E-cadherin, but and they don't all necessarily only contribute in different ways. So, there's very nice work from Holger's lab that I believe shows how E-cadherin is contributing to mechanical coupling of the cells. We also see that EpCAM is expressed, but EpCAM is primarily expressed in the cells that form the rosettes. And there is a whole biology of how EpCAM might be regulated and connected with MIR200 and various other factors that regulate EMT, and that there are so there are many different adhesion molecules. We don't know all of them, and they're all contributing in many different ways to both collective migration and to morphogenesis in slightly different ways. So this remains an area of of investigation that we don't know everything we'd like to know about yet.
Amazing. Virginie, we will come to your question. Just I'll take the question from Timothy asking, when CXCL12 was overexpressed or knocked down, do you see one cluster? Due to slowing down, or there is a couple of rosettes that form before the slowing effects are observed?
So our interpretation of that observation is that it doesn't matter whether you increase the chemokine or decrease the chemokine. In both cases, we observe that leading cells tend to slow down. And we are actually quantifying this more carefully right now. But the idea is that in those circumstances, the leading cells are slowing down, the trailing cells are continuing to move. And in that context, the preformed rosettes merge to form larger rosettes. In the case of the heat shock, actually in both cases, there is recovery, and that basically when the cells start moving again, you can often have a situation where rosettes that have merged together to form one large rosette, as the movement restarts, they will split to form two rosettes again. So I'm trying to think whether whether it happens most with the trailing ones or the leading ones, and I can't remember now for sure which it is.
Sorry,
great. Thank you for that. No problem. Virginia, do you want to ask your question? Thanks so much.
Thank you. Hi, Ajay. Thanks for
Hi.
I had a question following Arthur's question, actually, because I'm not sure I understood. When you say when you increase or decrease apical constriction or decrease cell adhesion, are you meaning experimentally or in your model?
No. So in the model, that's what happens, which is what surprised me. And so now what we're trying to do is use a bunch of manipulations. So the model predicts that if you only increase apical constriction, you should make smaller and larger number of rosettes. This was completely non-intuitive to me. I have actually a slide that explains, because I now understand at least theoretically why that happens, but we haven't yet. I was hoping to get the experiments done for actually today's talk, but we were not able to get those done. So we haven't experimentally tested this prediction. It's a kind of one of these intriguing predictions by the model. And, it's nice to make the model recapitulate things that already happens, but it's always interesting to see if the model makes a prediction that you had not anticipated and whether it turns out to be correct or wrong. So it's a test of the model, and we are still working on that.
And how do you, because you're talking now about your 2D pot model, right?
Yes, yes.
So you can't really test or model apical constriction, right? Because you don't have a 3D aspect there, and apical would mean.
Well,
what do you mean by?
So experimentally, for example, what we were planning to do is one of the experiments that seems a seemingly simple experiment, was to try to see what happens when you initially treat the embryos with blebistatin. What happens to over time to the configuration and size of the rosettes that are already formed. So actually, I'll show you something. Oops, here it is. So this is actually the simulation that showed that in the model, at least, if you increase the constriction strength, you get progressively larger and smaller rosettes. And actually, this can be understood in a rather simplistic way. So if this is a row of cells and they are all adhering to each other, and it takes some force A to separate or break the adhesion, and each of the cells is constricting and exerting a force C, then as long as this constricting force is less than the force that is required to break the adhesions, the cells will remain attached to each other. But let's say the force that the cells stick to each other is A, and it is 4 times C. Then if you have 4 cells constricting, eventually the force with which these cells will constrict will be adequate to break this adhesion. And so what you can see is that there is a relationship between the constricting force and the adhesive force that will determine whether the cells will remain attached to each other or break to form individual clusters. So in this context, for example, if I increase the constricting strength, then the number of cells that need to constrict together to be able to break this adhesive interaction will become less. So that's sort of a way of helping me understand this prediction that came from this model. And now we would like to see whether this prediction that is theoretical actually has some way of being manifest in the actual Primordium. I don't know if you understood this explanation.
I have to think about it.
Yeah. Yeah. I mean,
Do you wanna continue along those lines, or are you done with that? You look like you might still be thinking, Ajay.
No. So I mean, this is a sort of a theoretical explanation that helps me understand this phenomena that we found by playing with these simulations, and it makes sense to me. I don't know if I need to explain it again, but the bottom line is that cells remain attached to each other as long as the constricting force is not adequate to break the constriction, and that if C is less than A, well then as long as enough, so as more and more cells constrict together, eventually it adds up to the force that is required to break this. And so if C was 1/4 A, then once you have 4 cells constricting together, they would be able to exert the force that's able to break this, and that's what would determine where it would break and how many clusters would form in a given row of cells. I don't know whether this analysis is exactly correct, but it sort of helps me think about why we see this phenomena.
Yeah, great. Thank you for sharing that process with us. We have a question coming in from the audience on YouTube. Fang Lin is asking, have you looked at the ECM without skin? I don't know if that's something you can do.
I'm not sure. We haven't looked at. So what we did do was, after removing the skin, we tried to see whether we could whether just simply confining the Primordium was important. So we confined the Primordium with agarose. We embedded it with agarose, and the agarose did nothing. It neither got movement nor did it restore the formation of lamellipodia. Then what Damien did was to remove the skin and add matrigel. Now with matrigel, he was able to actually get some lamellipodia forming both in the basal cells as well as in the superficial cells, but he was not able to get effective polarization of the migratory processes, and he certainly couldn't get the Primordium to start migrating again. So in some sense, one line of investigation is to take the skin off and then try to put back things that would re-establish migratory behavior, and that would help us figure out the ways in which the skin is actually contributing. So we still haven't figured it out, and the skin may be doing multiple things. Besides providing an adherent surface, it may help to confine some of the signaling molecules that are providing, polarized directed migration. There are a number of different possibilities, and we haven't figured it out yet. It's just that we were surprised, given that the basal feet have the capacity to migrate on their own. The fact that when you remove the skin, well, actually I didn't emphasize this earlier in the talk, that the lamellipodia formed both by the superficial cells as well as the deep basal protrusions of the polarized cells are lost. So even though those cells are still in contact with the ECM, in the absence of the skid, their ability to make lamellipodia is lost, and they start forming these blebs, which was actually a big surprise to us.
Okay, awesome. We do have one more question in the chat, and it's from let me see if I can scroll to find it, from Humeng. And maybe Humeng, if you want to unmute and sort of clarify, please feel free to do so. So they're asking whether it's possible to generate a specific structure or morphology as an engineered method through the molecular pathway or genetic modifications, and then is following up with wondering about different cell types and how they together can generate designated structures.
Yes, hello.
So I'm not. Yeah. Do you want to clarify a bit? Go ahead, please.
Can you hear me?
Yes.
Yes, nice. I'm curious about thanks for talk and very nice. I'm curious about if we can control genetically or modified from the molecular level. If we can generate kind of a design structure, for example, I mean like the spheroid or like the more designed complex structure, we can generate what is the outer layer, what is the inner layer, and by just mix or just put the cells together to generate a design structure which is responsible from this level, from biological methods, for example, genetic modified or.
I mean, in some sense, I suppose it is possible. And in fact, the sort of simple cellular parts model that I showed just as an illustration is sort of an example of that. And I guess one could try to do that in a real system, because in that system, all you've done is taken to start with a completely mixed population of cells that are inclined to have different sizes and different adhesive properties. And you can, I just, when I made it, I played it around, and it self-organized to form that those structures that you saw in that simulation. But I imagine that this is possible. I mean, the other question, sort of related question you said about can you use a genetic tool to do something? The experiment that comes to mind is actually a very beautiful experiment done by Tatiana Piotrowski's lab, which also alluded to the fact that rosette formation is basically a very sort of mechanics dependent event. And what she did there was to create a line in which notch signaling was uniformly exaggerated throughout the primordium. So this creates a very interesting situation because for reasons I won't get into in detail, the activation of a notch in this very artificial way in all the cells, it actually erases all pattern within the primordium because it suppresses Wnt signaling, and you can actually simultaneously inhibit FGF signaling. And so you can erase all other pattern within the system. But in this situation where you have activated notch throughout the primordium, it spontaneously reorganizes to form one large rosette or two rosettes. And the idea is what Tatiana's lab was able to show, that when you increase notch signaling in this manner, what you do is increase the expression in the primordium of molecular factors that contribute to either adhesion or the formation of these apical like tight junctions and things like that. And so in a situation where you're uniformly increasing the level of these things that promote apical adhesions and constriction, you spontaneously generate pattern of rosettes. So which sort of illustrates that it's a self-organizing process that doesn't require a pre-pattern in the system, and it relies just on the mechanical properties of the that are resulting from the expression of various components of the cells.
Yes, yes. Because I noticed that as most of the person, I mean, biologists and the scientists are doing the research on the neutrally morphology genesis, and this is development biology is very fancy. But I mean, if I mean this is engineered method, do we have the toolbox or I mean toolkits for this kind of I mean before the experiment we have the simulation, or we do have um, some very clear mechanisms or which pathway or which molecular chemicals kit we can use to generate specific patterns or specific method. Do we have this uh, of like I mean do we have recommendations for this? I mean toolkits or repository of the materials. Do you have this kind of stuff?
I mean, we don't do it, and I don't know who specifically does it, but I am pretty sure that there are people who've been doing this sort of thing. I mean, the oldest stuff related to this is the whole idea that if you mix cells with different adhesive properties, that they will reorganize to form some kind of structure. This is a sort of an old idea that has been done both with modeling and with actually experimentally, and you can there are, I can't off the top of my head remember, but you can sort of take cells, dissociate them, and they will reorganize in many different contexts to form structures that were there previously. But I think the interesting thing in biology is that on the one hand, you have the sort of engineering aspect to it, that you can take cells, you can actually dissociate them, and because they have these distinct adhesive properties, they might actually be able to come together to form the structures that you recognized before you dissociated all the cells. But development works in a very interesting way, where it combines the mechanical the properties of the cells that would incline the organization of the cells in a particular way, but it couples it with signaling systems that set the stage, that allow one step to follow the next step to follow the next step. So it's so that in the end, development is this beautiful synthesis of combining self-organizing mechanical processes with a trajectory that is guided by self-organizing signaling systems that may provide patterns and a context within which the mechanical processes will happen in a much more organized, predictable, and a robust manner. And in a sense, that is what we are discovering in the morphogenesis of the lateral line, that there are mechanical processes that might naturally happen, but they happen in the context of signaling systems that set up the stage for those mechanical processes to ensue.
Yeah,
Thank you so much, Ajay.
Thanks.
Thank you so much, Ajay.
Beautiful summary. I just want to say that I loved that summary at the end. That was perfect and probably a great place to stop.
I thought it was also Ajay's pitch to bring everybody to do morphogenesis, which is amazing.
Yes, definitely. That's great. Yeah. Okay, I think I don't think we have any more questions in the chat. So I'm gonna stop the recording up on YouTube. So to everyone there, we've only got one more talk left. Sarah Wickstrom is next week, and then we're done for the season.